Welcome everyone to this virtual discussion on redefining the clinical utility of genomic testing in cancer care, hosted by the Personalized Medicine Coalition, where we will be introducing an updated definition of clinical utility and propose steps for its recognition and implementation. First, I want to thank our host, the Personalized Medicine Coalition, and all of our members and partners. The PMC is an education and advocacy organization based in Washington, D.C., with the mission of advancing personalized medicine. I also want to thank all of today's panelists, who I'll introduce in a few moments. And I especially want to thank all of you who have joined the webinar today, the audience, who share our interest on this topic and our goals in advancing personalized medicine. I am Daryl Pritchard, Senior Vice President of Science Policy at the PMC, and I will moderate today's discussion. I'm joined by three distinguished panelists to discuss the clinical utility of genomic testing in today's evolving cancer care health system. Today's discussion stems from an expert roundtable event held at the end of 2019 that was meant to address the definition of clinical utility of genetic tests in cancer care, which was hosted by the PMC and sponsored by Genentech. Special thanks to Genentech, for without them, none of this would have been possible and this webinar would not be going on. Especially Casey O'Neill, Alyssa Quinn, who is now with Blueprint Medicines, and Lori Lanphier, who raised the issue of the need for improved recognition of the clinical utility of personalized medicine and provided the support for the PMC to lead this initiative. Before I introduce the panelists, let me provide a broad background of the issue which we will discuss today. The clinical implementation of new health technologies depends on the utility that the technology brings to clinical care and the recognition of that utility. That is, the net benefits versus risks that comes from using the technology. Now, despite an expanding body of evidence supporting the clinical value of genomic testing, it remains underutilized in clinical oncology practice. So, facing challenges to its implementation, we considered what is the recognized definition of the clinical utility of genetic tests for cancer. And we considered this through the lens of multiple different stakeholders. The most often cited definition of clinical utility of genetic testing was proposed by Gross and Curry of the US Centers for Disease Control and Prevention in a commentary published in 2006 in Genetics and Medicine. This was the established and most recognized definition of clinical utility of genetic testing. The authors wrote, clinical utility in its narrowest sense refers to the ability of a screening or diagnostic test to prevent or ameliorate adverse health outcomes, such as mortality, morbidity, or disability, through the adoption of efficacious treatments conditioned on test results. A screening or diagnostic test, they wrote, alone does not have inherent utility because it is the adoption of therapeutic or preventative interventions that influence health outcomes. Thus, the clinical utility of a test depends on effective access to appropriate interventions. This definition is great in its context. In 2006, certainly was a comprehensive definition. It sufficiently captures a range of benefits and risks to clinical outcomes that derive from positive or negative test results of genetic tests that assess one or a few genes. However, it does not account for new technologies that have become prominent since 2006. Many technologies in personalized medicines, and importantly, genomic testing, nor does it address specific elements of utility related to cancer care. So the PMC called together this roundtable of experts, which included laboratories, providers, patients, payers, diagnostic and therapeutic manufacturers, and clinical guideline developers to update this definition relative to oncology. The updated definition the group developed goes as follows. The clinical utility of genomic testing in cancer care is the net benefit to cancer patients and health systems that is derived from applying information generated by multi-gene testing to screening, prevention, and treatment strategies that can improve healthcare outcomes, including through enrollment in clinical trials, facilitate shared decision-making, and reduce healthcare disparities. The utility of genomic profiling depends on its ability to provide information that is used to guide patients more efficiently to safer and more effective prevention and treatment strategies, as well as its ability to improve the practical knowledge base for health system decision-making. So with that introduction, let me introduce our distinguished panelists, all three of whom participated on the expert roundtable and will provide their perspectives on the clinical utility of genomic testing in evolving cancer care. First, we'll invite Dr. Lincoln Nadald to provide his perspective. Dr. Nadald is the Vice President and Chief of Precision Health and Academics at Intermountain Healthcare, a renowned integrated payer provider that is recognized as a leader in personalized medicine. We'll also invite Dr. Clifford Goodman to provide his perspective. Cliff is the Senior Vice President of Comparative Effectiveness Research with the Lewin Group and moderated our expert roundtable in 2019. And finally, we'll have Dr. Mark Stewart, Vice President of Science Policy at the Friends of Cancer Research, who will be able to provide a researcher and patient perspective of clinical utility in genomic testing. So let me turn it to you first, Lincoln. Lincoln, you are a long time standing oncologist and clinician and administrator of a personalized medicine program at one of the leading healthcare and oncology facilities in the country. Can you provide your perspective on genomic testing and clinical utility?
Yes, I'd be glad to, Daryl. Thanks for having me. And it's really a pleasure to be here with the other panelists. And I'm grateful, as you mentioned, for those who have logged in today and are our audience. And I'm looking forward to questions towards the end. I have lived this. I remember 10 years before this panel convened in 2019, I was at Stanford and I was living in this era where we were doing single gene testing. And I, at that time, met a patient, this 39-year-old woman who had 2 teenage daughters, and she had been diagnosed with gastric cancer. And many of you may be aware that that's a very difficult diagnosis. And I remember thinking and even talking with her about if we took the usual approach, we would get the usual outcome and she would probably not survive even a year. So none of us wanted that, including her 2 teenage daughters. And we decided we would try something unusual. And we did a multi-gene approach. In fact, we did whole genome sequencing of her tumor. It took quite a while to do that, but it worked. And to our great astonishment, we found a gene for which there was a targeted therapy. And we felt like that was something of a silver bullet for this patient. So we contacted the drug manufacturer knowing that this therapy was outside of her indication. And we requested to receive that, and it took additional time. We had to go through, compassionate use protocols and a variety of mechanisms, and ultimately the drug arrived. I'll never forget it. The day the drug arrived in our pharmacy, I contacted the patient and I said, "Guess what? This silver bullet therapy is here. I have high hopes. Your whole treatment team believes this is going to be effective for you, and you'll derive clinical benefit." And she said, "I'm so glad, because I haven't been feeling well lately. In fact, I'm sitting in the emergency room at the Stanford Hospital." And she got admitted to the hospital that day, and she wasn't well enough to receive the drug and never became well enough. She died just a few days later before she could receive that therapy. That was hard. That was a hard experience, but it taught me that there was utility here, or potential utility. So I took that learning, I transitioned to Intermountain Healthcare, where it's an integrated payer and provider system. And I thought, here we can implement precision medicine at scale. We'll have better assistance in getting targeted therapies for patients. And we embarked on that journey. And initially, we heard from payers a lot of no's, understandably. They were reluctant to approve or pay for the testing, let alone approve or pay for therapies. And we went to great lengths as providers. We were doing comprehensive genomic profiling in our patients with advanced cancer, and we were finding targeted therapies, but we couldn't always get them approved because the testing we had used was one of these multi-gene panels, and those were not broadly understood or recognized, even though they were laboratory developed tests. And we had to go to as far as having administrative law judge hearings. And I remember in the early days sitting on these phone calls with an administrative law judge and payer representatives, and the patients were there, and we would describe the situation, and it was emotional and it was stressful, and those usually got approved. And so after those first couple of years of hearing a lot of no's and going to great lengths, things began to relax and we began to publish our data. And I think all of that helped. And our data suggested that when we used comprehensive multi-gene testing in patients with advanced cancer in a big health system, that we saw a decrease in overall healthcare costs. In fact, our exact data were that we found $700 of savings per patient per week of survival. So if you do that over thousands of patients over a year or more of survival, it really adds up. And furthermore, patients lived longer. They lived twice as long. So we published that, and subsequently we have had much better success in not only getting the testing paid for, but also therapies approved. So I share that evolution and that journey to illustrate really how this whole field has evolved. And you go all the way back to the early 2000s, where we knew about a handful of genes and we would test for those in melanoma or in lung cancer, to today's world where it would be unthinkable to treat a lung cancer patient without knowing about the 8 or 10 or 12 biomarkers that are FDA approved in that indication. And we even have tumor agnostic biomarkers that exist now, with tumor mutational burden and MSI status, and there's a variety of others. So we live in a world where it's widely understood that multi-gene testing provides clinical benefit to patients, and yet we haven't taken the effort or the steps to really update all of the underlying definitions. And that's what this roundtable was about. That's what this meeting is about. And that's what the manuscript is about that we published recently. It's an update of the clinical utility associated with this approach to precision oncology. And it's warranted and it's time. And I'm excited to have been a part of it.
Thank you, Lincoln. Now let's turn to Cliff Goodman. Cliff, you're another recognized expert in the field and also sat as the moderator of the roundtable discussion, which involved all of these different perspectives. What's your take on clinical utility and genomic testing in cancer care?
Well, thanks, Daryl. As the facilitator, I'll do you one better. Rather than my views, I'll reflect and share with you the views of the expert roundtable. And so it's interesting, as you heard Daryl mention earlier, the expert roundtable considered that the current definitions, including Gross-Curry and some others, of clinical utility and genetic testing do sufficiently capture a range of benefits and risks that derive from positive and negative test results, especially assessing one gene or a few genes. So it's not like they were knocking that down. They're just saying that definition is as fine as far as it has gone, but maybe fading a bit in relevance. So let's take a look at key aspects of the current definitions. Just keep this in mind for the sake of making a compare and contrast. The current definitions tend to look at individual patients as opposed to, let's say, populations. They do indeed look at outcomes, reducing mortality, morbidity, and disability, so that the utility aspect does go all the way to outcomes, not just analytic validity and clinical validity. It does link clinical utility to the adoption of efficacious treatments. That's fine. And a really nice aspect, especially of the Gross and Curry definition, is that it does keep in mind that clinical utility isn't really present unless you know you can get the effective and safe treatments. If you don't have access to these, then where's your clinical utility? So those are as fine as it goes, a very nice basis. But through its deliberations, the group thought through, well, what might an expanded, contemporary, future-oriented definition encompass? Well, it would certainly encompass improved clinical outcomes, no argument there, but maybe perhaps the potential for a broader set of outcomes over time. So thinking more broadly about potential outcome improvement. Another aspect that the group brought up was, let's get beyond the individual patient. There are to be population benefits and health system benefits associated with the data compilation and analysis and more efficient sample processing. you can do a lot more with a lot more data because of we can manage large data sets more efficiently. The analytics are better. And beyond looking at an individual patient, there are health system benefits and population benefits as we accumulate these larger data sets, whether they be through genomic testing or other sources of data. Another one was for opportunities for shared decision-making. Let's think about what the data set or the information basis is for shared decision-making. These newer tests can bring more to the table about risk factors, whether they're genomic or other. There's a lot more data to be brought, information to be brought to enrich real-time decision-making with clinicians, patients, and their families. So that's a third aspect. And then another one, which was really interesting, it had to do with patient access again, but to broader prevention and treatment strategies, including the aspect of, can we bridge clinical utility from what is known about presently approved treatments, let's say, bridging that when sometimes in oncology they don't work, into clinical trial enrollment. So one of the aspects of clinical utility here is, where do you stand to improve if current first and second line treatments don't work? Can we bridge you smoothly into clinical trials, which of course the research, keep that in mind, but that is another avenue for clinical utility. This was a really important point I thought that the group brought up. And so what then does the updated definition bring to you? Certainly improving healthcare outcomes in the broader sense, as I just mentioned. Certainly the potential to enroll in clinical trials, which may improve clinical healthcare outcomes. Certainly facilitating a higher level of shared decision-making, more informed decision-making. Interestingly enough, also incorporated in the new definition was reducing healthcare disparities. Why? Because healthcare disparities, which are increasingly important and well recognized throughout the healthcare sector, certain risk factors associated there may be picked up, perhaps, in genomic testing, but not even just genomic testing. What other sources of data, social determinants of health and others, can be brought to bear? These multiple data streams, including from these genomic tests, into that crucible of decision-making. And then finally, enhancing and enriching the knowledge base, the practical knowledge base for health system decision-making. So I hope what I've done just there is kind of show you where the expert panel said, okay, the current definition is good as far as it goes. Here's some things that need to change. Here's a new proposed definition, and here's how they contrast. Back to you, Daryl.
Thank you, Cliff. That really does set up nicely and lay out the conversation that we had and that we intend to have today. Let's turn it to Mark Stewart, Dr. Mark Stewart with the Friends of Cancer Research. Mark, your organization is another leader in the field, especially in cancer research and personalized medicine, representing both the research and patient perspective. What is your perspective on clinical utility genomic testing in cancer care?
Thanks, Daryl, and thanks for the opportunity to participate in the discussion today. I think before maybe I get into clinical utility, I think it's worth just reflecting on how far we've come over the past few decades, and really is kind of the impetus, I think, for this discussion. I think we've truly seen some substantive advances in our understanding of cancer biology, and I think that's had a tremendous impact on cancer drug development and technology that's being implemented in patient care. I suspect many of the people that are joining us today certainly have context into this, but it's worth noting that, over the years, we've seen an increase in 60% of the number of oncology drugs in clinical development, and a doubling of the use of biomarkers to help identify patients likely to respond to those therapies. And in fact, predictive biomarkers are associated with roughly 60% of new oncology drug approvals. And so genomic testing certainly isn't going away, and in fact is likely going to become an increasing role in oncology care and the patient journey. And I think it's worth kind of reflecting on what that biomarker testing journey looks like for a patient, which starts with the suspicion of or suspected case of cancer, which usually leads to some form of biopsy, leading to either confirmation of cancer, but in parallel, hopefully some genomic testing. And so with that, comes some interpretation of those test results and decisions to be made between the patient and the physician on whether that indicates a specific treatment should be used. And that's not the end of the journey anymore. Like I mentioned, there's a handful of drugs now available, and often and there are moments where progression or recurrence might occur, and so being able to rely on those biomarker testing and genomic test results to help navigate that next series of decisions that need to occur is quite valuable. And I think that's one important component to acknowledge here, is that the genomic tests that we have that are able to detect multiple different mutations might play a role at various aspects during the patient's journey as they're potentially going from one treatment to another. And I think, as Lincoln had mentioned in his opening comments, there might be moments where there isn't currently an approved agent, but ongoing clinical trials and these genomic test results can hopefully open the door to those options as well, if no alternatives are available for the patient. And so with the uses of genomic tests, I mean, how do you begin to measure their usefulness or clinical utility? And I think there's some criticism, around clinical utility, and that it's a very broad term that's used to denote the usefulness or value in clinical practice. And unfortunately, I don't think there's a singular or agreed definition, and demonstrating is certainly not straightforward. So I think, the conversations that we've had as part of this working group and putting out this definition hopefully can provide some alignment and greater understanding of what those components are that can help inform clinical utility. And I think another important, component to consider here is that I think the kind of maybe the varying views of clinical utility is likely driven by different views of what's considered important. And so perspective is certainly important. And, I think a test is typically considered to have clinical value when it provides information which can be used for clinical management decisions. And that's obviously an important factor for healthcare providers and systems. But these results, also have further psychological, social and practical implications, for patients and their families. That's also important to measure. And so moving beyond strictly just clinical questions to include kind of practical concerns, such as organizational issues and implementation and feasibility of conducting these tests and cost effectiveness, I think provides a more holistic view of the usefulness of genomic tests.
Thank you for that, Mark. And thank you to all of you for excellent setup for the conversation that I hope we can continue to have. I'm now going to proceed with some questions and answers. And as we discussed before, would love this to just be a conversation about the clinical utility and about how to move forward with genomic testing in cancer care. So I'm going to open it up, starting with a question that Mark really just laid out for us. As you mentioned, Mark, clinical utility is a broad term, and there are many definitions, and some are recognized as strong and some are weaker, recognized as weaker. But a key to that are there many different viewpoints on clinical utility. For example, a payer may have a different outlook on what the clinical utility of a genomic testing platform is than a genomic testing manufacturer or a pharmaceutical manufacturer or a patient. So the question that I'm going to pose to all three of you to start with is, why is it necessary to update the definition of clinical utility that the definitions that we had been looking at in cancer care, and to what end? To what end is this practice of thinking about the definition going to help move the field forward?
Well, Daryl, this is Cliff. Maybe I can get it started insofar as sort of the why, and then I suppose Lincoln and Mark can tell you what that gets you. And again, reflecting the views of this diverse expert panel, if we look back at our notes and some of the contents of the manuscript, a whole set of things has happened. What has transpired since earlier definitions were put in place? Well, for one, multi-gene assessments. These very large testing arrays absolutely changed. In fact, they do disrupt what testing looks like these days. So the technology is itself orders of magnitude more powerful. Also, something that's changed that's relevant here is greater diagnostic processing efficiency. You can do more faster and ultimately cheaper, it is hoped. And for example, if you want to get technical, the biospecimen conservation is greater compared to running multiple single gene tests. And we hear a lot about that, the biospecimen conservation being so important and precious. And so with this new technological approach, you get a lot more out of your specimens, essentially, which allows you to do what? You get a lot more up front. You can now come back and look for additional biomarkers later on. Okay, so you grab a lot more data up front. It's well conserved. You may have questions that crop up later on that allow you to come back to that data set. Another thing that came up was the potential for drawing from linked databases. So once again, let's not think just about genomic testing in these multi-gene assessments. Let's think of all the data sources that are flowing together. Think of the potential. This is what the expert panel kept pointing out. If we can link claims data, administrative claims, and electronic health record data, sometimes even through natural language processing, clinical notes, and so forth, registries, other patient-sourced data, data sources on social determinants of health and so forth, along with the multi-gene assessment arrays, right? So these multiple flows, convergent flows of data, provide a much greater potential for what can be learned toward in the direction of clinical utility there. And of course, advanced analytical tools, we can do a lot more faster than ever before, so the yield is greater. And then another thing that the group kept bringing up was this, and I think it's sort of a broader social progression, that is, as years go by, we all, as patients and family members, seek greater patient autonomy. I want greater autonomy for myself and my family members, and in order to be more autonomous, I need more data. I want to know more about my risk factors, genomic or otherwise. And so those are like 5 or 6 things that the group noticed had changed, had transpired since the context or environment in which the earlier definitions were developed.
You know, Cliff, I love that point about patient autonomy, and I want to just pick up on that here for a minute. That's part of the fourth component of the new definition that is proposed right from this group, which is shared decision making. Another way to say patient autonomy. I see this all the time. Patients want to know if they're going to start a therapy that there's some reasonable chance that they'll benefit from it. And conversely, they don't want to start a therapy, none of us would, where there's no evidence that it's really going to help them. And that's where today's world is a little different than it was, 15 years ago. And so when we do these multi-panel tests, multi-gene large panel tests, we identify not only predictive biomarkers, as Mark so nicely outlined, but we also identify prognostic biomarkers, and we identify biomarkers that are both positively and negatively predictive. So we can appropriately help a patient avoid a therapy that we now have evidence is not going to benefit them and in fact may harm them. And so, then patients get to participate in a more fully informed way. The other thing that I've seen this really do is it allows patients to really feel like they've done everything they can. I mean, we've all heard of scenarios, and I've personally walked many patients through the scenario where they've tried everything, quote unquote, and they want to feel like they have turned over every stone for their mother or for their father or for their loved one. And that's kind of what this allows, right? Is you can say, we have done everything we can. I mean, we've mapped all of your genes. We've assessed you for all possible clinical trials. There aren't other options. You really are making the right decision here by making a transition towards a different approach, whether that's, end of life care or whatever. So it's that patient autonomy, it's the shared decision-making that becomes so valuable. And I have other experiences about how this benefits health systems that we can talk about as well, but I just wanted to highlight what you nicely laid out, Cliff.
I think that's an important point just to carry forward a bit around that shared decision making and ensuring that patients have the available tools and information also to contribute to decisions around what their treatment plan can look like. I know many of us have had to deal with cancer in some fashion in our lives. And for me, it was helping my dad navigate his care options when he was diagnosed with cancer. And while his prospects were bleak, I mean, he certainly wanted to have the best quality of life for as long as he could. And that meant finding the right treatment with the right balance or absence of toxicities in his situation. And I think having, the results of these genomic tests, as Lincoln had mentioned, and Cliff, provides, numerous amounts of information, not only to help, understand what types of treatments are available, but maybe which treatments are likely not going to be beneficial, and additionally provide insights into, prognostic factors around what outcomes to expect. I think an optimal output of a genomic test is that it's opening doors for patients, that it's providing access to some therapy that will provide benefit. But again, there's additional information that one can glean from these tests, such as indicating which therapies to potentially avoid. And just wanted to pick up on a point that Cliff had made earlier, passively, recognizing the wealth of information that these genomic tests are able to capture, not just around mutations that we know are to be actionable, but other mutations that aren't maybe actionable today, but will be tomorrow, and how this information can help contribute to that ongoing research. But with that, though, it does require that the systems be in place to where you can actually, gather that information in a meaningful way. And I think, Intermountain Healthcare is certainly a good example of that, but I don't think that's the norm across the US in terms of these learning healthcare systems. And I think that's something that likely needs a bit more attention to ensure that we're not losing this valuable information in the ether, but it's being collected in a way where it can be interrogated and help future patients.
Mark, that is a great point. And, I've seen this develop. One of the first things that we implemented when I arrived at Intermountain was a molecular tumor board. Everybody's probably familiar with these, right? They're patterned after the traditional tumor boards where you get a bunch of experts in a room and you review a patient's case, and with multiple minds on the problem, you can come up with a nice solution that is helpful for the patient. And molecular tumor board is a similar idea, except it's an emphasis on the genomic and other molecular profile of that tumor, and you're really looking for the right treatment based on that broad additional information. And so, Mark, what you described, where we have learning health systems, is so important. And molecular tumor boards definitely, all over the country. I have had a nice opportunity to sit on molecular tumor boards from the East Coast to the West Coast, and all of them depend on what they've learned from previous patients and on the collective knowledge that's published in the literature and on their own institutional experience. So what you just said about, capturing additional information and that informs the treatment plan for the next patient is true. That definitely happens. And Cliff, I felt like that part of that came up in this group, right? There's that recognition. Experts all over the country, the leaders in this space, know that it's about learning as much as we can, and we apply that to the next patient. And tomorrow's patient is going to benefit from yesterday's patient's experience. I mean, that's a principle. That's a foundational principle of medicine.
So I'm hearing a number of themes that arose that Cliff kind of laid out and then were corroborated by Lincoln and Mark. on the clinical utility around information management, more information. Cliff also mentioned efficiency in care and that access issue, especially as related to shared decision-making or getting the patient more involved. Let me just highlight another point that Cliff made, only that hasn't been expanded upon, and see if we can get a little expansion upon this. And that's the building of this knowledge base for future scientific discovery and how the use of genomic information can be pooled. The information can be pooled and then used perhaps to develop further validated biomarkers and diagnostic tests that can drive additional clinical utility upstream down the road. What do you think the role is for that learning health system in all of this?
Well, I'll start it off, Cliff here again. As we've alluded to before, genomic testing is filling a large data set, multiple large data sets, along with other inflows of information from different sorts of data. If you are, for example, large integrated health network, a large integrated delivery system such as Lincoln's, you're starting to get a population size big enough where for many conditions, you've got multiple streams of data that are providing significant amounts of insight. So someone in a large integrated delivery network can say, in our network's population, we have seen patients like you. In other words, patients with similar indications, comorbidities, demographic factors, genomic data, social determinants of health and other demographic data, where you can say there's a population, a real world population for whom we have genomic as well as other data sources that we can apply and use to say, there is a knowledge base that may be relevant to you because you have a lot of similarities to that particular cohort because we've got enough data to do that. And some of the larger payers, they're also saying, gosh, we have millions or tens of millions of beneficiaries and people on our plan. So you're at the point where they were saying, we're getting large enough cohorts of data for populations and subpopulations to start learning more than we could before. Again, given the analytics and so forth. So multi-sourced, multi-gene assessments are one kind of supply line to these larger data sets, to the extent that you can link the other data sets we mentioned, and we have larger healthcare delivery systems, the population, the potential to improve population health continues to rise.
Cliff, such a great point. The other phenomenon that happens or is enabled, the other real opportunity that's enabled by this aggregation of data is it allows us to measure our performance against known gold standards. So we just heard Mark cite that, 50 to 70% of new therapies are precision therapies and have, associated biomarkers. And at the same time, recent publications suggest that maybe as few as 30 to 40% of patients in the United States with advanced cancer are receiving the appropriate kind of biomarker testing, right? Not even the therapy, just the indicated biomarker testing. So we know that because we've been aggregating these data sets, and it allows us to self-reflect and say, why are we so far behind even what are approved biomarkers? How come we're not testing patients appropriately? And it's largely because there is an education gap. And so that's part of what this definition of clinical utility does. I think it emboldens other policymaking and guideline making entities to continue to update their guidelines, which then allows physicians to align their practice appropriately. Changing physician behavior is the hardest thing in all of medicine. I mean, it is really challenging. And so it takes, a comprehensive effort like this one and many others, so that, the evidence is so momentous that physicians then change behavior.
Lincoln, you just reminded me of something I wasn't going to say, but it comes up now, and that is this updated definition provides a little more focus or highlight to one or more of the problems to which you just alluded. One is, are people who are indicated for a multi-gene assessment getting it in the first place? Are we recognizing that they're indicated for this. And that can be physician awareness, system awareness, even patient awareness. And not everyone who's clearly indicated for a test is getting one. When they get them, when they get the tests, are they run in a timely basis? Yes, no, not always. And when they're even run on a timely basis, do the results come back in time to inform the shared decision-making? And sometimes it's a little late. You kind of missed the bus. The bus has already left. And so think of the unmet potential here, just to kind of do the things we know are indicated for individual patients. So that's a systems issue, but there is unmet potential here that, again, our experts and from different stakeholder perspectives expressed at the meeting.
You know, to maximize utility, there are really two things that have to happen. First of all, a patient needs to have the test ordered, right? So physicians, providers have to order those tests. And secondly, it has to be done at the right time. It really doesn't do any good to wait until the patient is at the end of their rope before you order this, right? Because then by the time you get the results back, too often they've declined and they're no longer a candidate for that key clinical trial or that key therapy. It really needs to be ordered at the time that their advanced disease is diagnosed. And I would love to see payers recognize this utility phenomenon and start to insist that if you're diagnosing this patient with with some kind of advanced cancer, we insist that they get the right kind of testing. It actually benefits payers. Those patients, it'll cost less for those payers if the patients have had the right kind of biomarker testing, and they'll do better. They'll have fewer side effects, they'll have fewer hospitalizations, and they'll feel better. They won't need as much supportive care if they get this kind of biomarker testing and the right kind of targeted therapy. So it's time, I think. Historically, payers haven't really played a leadership role, and that's partly on purpose, right? It's just not their mandate. However, they do play the role of adoption. And I would like to see payers adopting this more broadly. It's okay for them to, evidence is there.
Not to be too repetitive, but I think going back to the biomarker testing journey for a patient, where it starts with a suspected cancer, to getting a biopsy, to the testing, to interpretation of the results, and hopefully matching to a therapy. At each of those areas, there's potential pain points that have been highlighted. I think issues with tissue or biopsy acquisition, biomarker testing not being prescribed. And then when testing is done, maybe it's not a multiplex test. It's just a test that's looking for a single mutation, and so you're getting limited information. The turnaround time. And then when you do have the results, are they being interpreted correctly? Are the treatments being started before the test results are even received, which is often a reality? And then are patients even being told the results? Are patients receiving the right treatment based on those results? And so if you just take a traditional measure of utility, hopefully I'm not oversimplifying it, but that the test is leading to improved outcomes, you're going to lose, some of this granular information along the way that's ultimately going to impact whether that test has utility or not. And it really gets at the behaviors, and I truly believe that this proposed definition kind of captures that. If we can begin to measure those clinical behaviors and the impact that those can have, I think you get a fuller picture of kind of what's happening along that journey.
Mark, thank you for bringing up those points and for laying out a follow-up, a bit of work that the Personalized Medicine Coalition is doing. We're doing just that. We are measuring the impact of all of the different clinical practice gaps that can occur throughout the precision oncology pathway. And we're doing it in lung cancer because that's where we have the most data. And without giving too much away, we're about to submit the research report for publication. But we found that only about 34% of eligible patients in lung cancer have made it all the way through all of those different points that you mentioned to get an appropriate targeted therapy who are responders for it. So we're losing patients at many different stages. They never get testing at all. How many, what impact of the population are we losing to that? Where are we having biospecimen processing problems? When is the turnaround time for the testing of not allowing the physician to wait to get testing, and then downstream, when even though there are results indicating a targeted treatment, are they not getting because those results haven't been applied appropriately? So we hope to do that. And I guess the key thing, and we will be, and just to show, to mention that only 34% of patients are making it through the precision oncology pathway in lung cancer, where precision oncology is well more established, is an indication of the problem in all of cancer. And I think one of the key things that we are trying to accomplish here with this recognition of this definition of clinical utility is to help address some of those problems. Let's make sure that at every stage, all of the stakeholders that are working towards that patient getting the best treatment possible are aware of all of these benefits and risks of genomic testing.
Yeah, you know, Cliff, that's an interesting phenomenon, right? So 34% of patients in lung cancer are making it through the entire pathway, and I suspect lower in other cancer types, given the establishment of precision oncology in lung cancer specifically. And that's a little disconcerting if you think about some of the biomarkers that have tumor agnostic indications, such as tumor mutational burden or MSI status or NTRK fusions, et cetera. It's increasingly hard to justify if you were treating a patient with an advanced cancer and you had not checked those biomarkers. I don't know what the explanation would be.
Yeah, there is no explanation except for, awareness, education, and standardization of process. So.
Well, that reminds me then, once again, insofar as alignment of stakeholders and awareness, once again, and I'm actually kind of in my mind's eye looking at the room of experts around the table here and what they were saying, everyone was aligned on certain things. Everyone wants better outcomes, all the stakeholders. Everyone wants better informed shared decision making. Everyone wanted greater test accuracy. No one was against, well, let's put it this way, everyone wanted fewer false positives and fewer false negatives. Everyone agreed that false positives lead to unnecessary and sometimes even harmful interventions and the cost that those accrue. So that was certainly a big body of agreement or alignment with the group. There was lack of alignment, or at least progress or maturity on education and awareness with some inherent variations. As we just heard, some clinicians expressed not having enough real-time information about tests, running tests, test results to support their decision-making. And so there was disappointment there on the part of clinicians and sometimes even payers as well, and certainly patients, as you heard from Mark just now. Now, two really interesting issues, one especially for payers, and this may have been the thorniest in the room, and that was even where payers agreed with the other stakeholders and the things I just mentioned, payers did say, listen, we have some constraints in coverage policies, particularly when it comes to interpreting our, i.e. the payer speaking, reflecting what they said, our interpretations and criteria for medical necessity, our definitions and criteria for what is experimental or investigational. And so to some extent, they said our hands are basically tied. These are the ways in which we have to make our coverage decisions. And I think many in the room agreed that the historical basis for those was something that may not be as current as we'd like. Why should payers pay for something that's investigational experimental? Is that in the interests of beneficiaries, of their company, of those who pay premiums and so forth? So that's something that they were expressing may be tying their hands just a bit. Now, there are some provisions in place, some policies like Medicare has coverage with evidence development, okay? And Medicare and other payers have provisions for paying for certain aspects of clinical trials. So it's not like you can't ever pay for something that's investigational, for which the evidence base is not totally locked down. So there are some instances or models of openings along the way, but that is certainly a major issue here. And even though this group proposed an updated definition of clinical utility, the decision makers out there are subject to certain policies that may still be based at an earlier time. I think that's the way I want to say it. Now, one more related thing, we had people that developed clinical practice guidelines. And I will say, and it was a good representation of that group, and I have my own kind of professional involvement in clinical practice guideline development. And that is, they were increasingly familiar technically with, I know what analytical validity is and clinical validity and clinical utility. They're up to speed on that. They know what kind of evidence is needed, even for diagnostic accuracy and the kinds of tests and study designs that work there. But what they're also aware that the evidence base for clinical utility of tests, up into including multiplex tests, is not the richest. It's pretty sparse. And so if what you're trying to do is develop a truly evidence-based clinical practice guideline using rigorous studies and trying to seek out evidence of clinical utility of screening or diagnostic tests, you may not find a lot. So that highlights a gap that needs to be filled if indeed we want to pursue this more current definition of clinical utility. That's an important thing not to miss. And the practice guideline folks know that. And by the way, payers often cited that, well, we look to the clinical practice guideline developers to help guide our policies, our coverage decisions as well. So those are interlinked.
You know, they are interlinked, and it's an interesting situation because on the one hand, we'll hear payers, as an example, say, well, we're going to approve therapies that are consistent with national guidelines. And at the same time, those same national guidelines indicate for multiple biomarker testing in those different disease states. And yet those same payers don't insist on multi-biomarker testing to be completed. So I think, you know, we all suffer from this at times, right? We need consistency. And so it's just, I think it's an educational gap. I even think that gap extends to entities with whom payers contract. At times, payers will contract with expert advisory groups that help them, you know, discern between all of these different tests, because it's a little overwhelming for, you know, I'm not trying to pile up on payers, but it's just sort of here in front of us right now. To stay up to speed on all these different tests, not just in oncology, but outside of oncology as well, it's a lot. And so they'll often contract with third parties to help them discern between those. And so now we're reliant on those third parties to stay up to speed on the rapidly evolving guidelines, right? So as NCCN and ASCO and others update their guidelines, are we sure that those new recommendations make it all the way to not just payers, but also to the contracting entities that the payers rely on?
It's hard to catch up with the science every day, Lincoln.
Yeah.
Well made. It's moving quickly.
So we only have about 5 minutes left. I can't believe it when I looked at the clock, how robust this conversation's been. We've only gotten through half of what we've hoped to have covered, but it just shows the enthusiasm and need and necessity for us to continue to drive these concepts forward. But let me ask one more question, which I think will take us a full 5 minutes to answer, and that is, how could the new definition that we proposed be integrated into health practices with attention to the needs of all of the different stakeholders and decision makers that we've been talking about? What can we do to make this, to implement this definition?
Maybe as an initial start, I keep going back to the patient journey, but each of those kind of decision points or actions that occur, there are data elements that likely can be captured, both around the behaviors and the decisions that occurred, and I think ultimately will help provide a greater glimpse into the utility of these tests. I think, you know, Lincoln and Cliff can likely, you know, add a bit more detail, but I think it's from what, one thing I've taken from this conversation, from the working group discussions, is that again, perspectives matter, and I think it will be important for those that are trying to capture data to understand the utility of a test. You ought to engage patients, physicians, payers very early on, and to understand, have understanding of what from each of those perspectives matters, and then making sure that you're designing your study design in a way to where you can actually answer and address those questions. I think if we're saying that a value of genomic testing is its ability to contribute to research, I think then you need to demonstrate that you have the infrastructure and capabilities to actually make that happen. And we touched on that earlier. And I think while it's, coming up with a common definition, gets us in a good place, I think, next steps really ought to be how then you implement that definition, and what are those important data elements? How can people begin to capture those outcomes? What are informative study designs? And what are pros and cons of different data collection methods? I think having alignment there will also be helpful to enable people to actually operationalize the definition.
Yeah, that's well said, Mark. I would just speak, Daryl, and answer your question. I'd speak to three key stakeholder segments, to patients and patient advocacy groups. I would say, if you're diagnosed with, or your constituency in the advocacy groups are diagnosed with an advanced cancer, ask your provider, am I a candidate for comprehensive genomic profiling? Am I a candidate for broad genetic testing? I think every patient should be asking their provider that. To providers, I would say, when you're treating a patient, when you have diagnosed a patient with advanced cancer and you're going to begin treatment, you should ask, why haven't I ordered comprehensive genomic profiling? It should be the exception. When you haven't done it, it should be the exception. Your default should be to order it, in my opinion, in order to be compliant with guideline-based biomarker testing. And thirdly, to payers, I would say, is it time for you to start demanding that patients have biomarker testing? We demand it for all kinds of therapies. We ask for prior authorizations, and I think those prior authorizations should include, did you do a comprehensive genomic profile? Because the evidence suggests that those patients do better.
Lincoln's comments following on Mark's have tweaked my response a little bit. I'm going to go back to what our expert panel was saying insofar as how do we move next? And it was this. This roundtable is not held in a vacuum. Folks brought their perspectives and attitudes and observations about the broader healthcare system in which we live. And so this updated definition of clinical utility is not placed on the table in isolation by itself for you to consider whether we like it or not. This updated definition of clinical utility is harmonious with our advanced data collection capabilities overall. We are collecting data from so many other sources and linking it and applying advanced analytics in such a way that how can you not want to take full advantage of what multiplex gene testing has to offer, even unrealized potential? It is another data flow into multiple data flows, all of which are advancing our ability to provide real feedback, real world data to patients in shared decision-making, as well as advancing population health and health system efficiency. So it's kind of the current thing to do to complement everything else that's going on in our healthcare system with regard to assembling data into evidence, information, and knowledge.
Well, thank you for that, all of you. And I agree. I think the overall conclusion is genomic testing in cancer care, its clinical utility, we can't afford not to. This is the science and the technology dictating where we're going. And I really appreciate all of your perspectives. Thank you, Mark. Thank you, Cliff. Thank you, Lincoln, for participating in this discussion. And once again, I want to thank the entire audience. And if you have any follow-up thoughts or questions, any of you, please feel free to reach out to the Personalized Medicine Coalition, myself in particular, if you want. D. Pritchard at personalizedmedicinecoalition.org with your thoughts. And with that, I think we're going to wrap this conversation and continue to work together. Link, Cliff, Mark, I look forward to continuing to work together with you to move this definition forward and to really impact clinical cancer care. Thank you all.
Thank you, everybody.
Thanks.